Okay. All right. Thanks, Alex and Randy, for having me out. Really appreciate it. I've been hearing about all the amazing things that you're doing down here at ASU and finally have a chance to see it firsthand. It's been fun. I've had a great visit so far and really appreciate it. So what I'd like to talk about today, let's start off with this phrase, you are what you eat. Everybody knows that. And I think it has an intuitive appeal to it because we are physically built from the molecules that we bring in from the food that we eat. And so if you're eating this stuff, you're built of more saturated fats, you're built of more cholesterol, sodium, your health is going to suffer as a result of that. But there's a second meaning to this because it's also the quantity that we eat, right? And if we eat more than we expend on a regular basis, then you end up gaining weight. We have an obesity epidemic here in the United States, but it's also happening globally. So you're seeing body mass indexes going up. And not surprisingly, those diseases that are associated with that also are going up. So this top line here is cardiovascular disease. You see it's the leading cause of death globally, and it's trending upward. Now, this association between eating too much of the wrong things and cardiovascular diseases is so well established, like it's so intuitive to us at this point, that when data like these were first reported about 25 years ago, people, I think, were surprised and also a bit skeptical. What this shows is risk of heart attack not on the basis of your adult weight, but on the basis of your birth weight. And you can see it's the lower birth weight individuals who have the highest risk of heart attacks. And this happens to be from Britain, work by David Barker, who sadly passed away at the end of last year. But I think he, more than anybody, has helped promote this idea that the roots of adult health trace in part to experiences during intrauterine and early postnatal life. And we've known for a long time that when a fetus is stressed or undernourished, it's going to slow its growth rate, and as a result of that, you're going to get lower birth weight. What's new and what David proposed and what I think is mostly been confirmed by a lot of 20 years of research is that there's a lot more that's being changed in utero, okay, biologically. You're getting changes in lipid metabolism, glucose metabolism, where on the body you prioritize fat deposition, blood pressure regulation, inflammation, other things, right? So these are all changes that occur and persist into later life and increase risk for cardiovascular disease. Now, here's another example from a human population, also from Barker and his colleagues, showing lower birth weights associated with higher rates of diabetes. Everything so far I've shown you is based on birth weight, which is obviously a pretty crude marker of fetal nutrition. A bigger baby did receive more nutrients, and in that sense it is a marker of nutrition, but they're also clearly genetically small babies, genetically large babies, and so on. And because of that, animal model research, where you can really experimentally control one thing and kind of nail one exposure, has been really important in this field in validating this model. So this is one example. This happens to be a guinea pig model. And these open circles here, this is insulin, fasting. This happens to be the fasting state here. These open circles are individuals whose mothers had their nutrition restricted by 30% during pregnancy. And you can see that under fasting conditions, their insulin is much higher. Then they get a glucose infusion, much like you would get at a doctor's office as a test for diabetes, and their insulin shoots way up above the control animals. What this shows is that in these prenatally undernourished animals, they're having to work a lot harder to clear the same amount of glucose. They're having to overproduce insulin. This shows us that they're insulin resistant. Their muscle is not responding to the insulin. And that's the primary condition that eventually leads to diabetes. Right? So when we see these kinds of crude associations between things like birth weight, this very crude measure, and higher diabetes risk in humans, we think that part of that is a causal effect of fetal nutrition, in part because of these kinds of animal model studies. All right, so all of this then leads to a kind of a modification of that old idea. Maybe it's not just what we eat as adults, but also what your mother ate when she was pregnant with you that matters for your adult health. And some of us have been studying this for a long time. I think it's only gradually been percolating, into the press and into the popular media and so forth. And here's an example of that. A couple of years ago, Annie Murphy Paul wrote a book called Origins that kind of explored this literature and talked about it. And this is a piece that she wrote for Time, How the First 9 Months Shaped the Rest of Our Life. Here's just one quote of it to give you a feel for how this is being interpreted. Much of what a pregnant woman encounters in her daily life, the air she breathes, the food and drink she consumes, the chemicals she's exposed to, even the emotions she feels, is shared in some fashion with her fetus. The fetus incorporates these offerings into its own body, makes them part of its flesh and blood. So the perspective that you get out of a quote like this is sort of like anything that the mother does during pregnancy is going to have some direct effect on the fetus. And you can imagine that if nothing else, that's going to raise a lot of anxiety among expectant mothers. Now, luckily, this is a simplification, and it's not quite that simple. Some things do, in fact, have fairly direct effects on the fetus. Some dietary, some things that you eat do have those effects, while other things do not. And so let's look at that. What effects do the mother's diet during pregnancy have on fetal growth and development? All right, some do matter. So we've known about this for a long time. Heavy metals, things like lead and mercury, have bad effects. There's been a lot of press about things like BPA, which can have negative effects. Heavy alcohol consumption, drug consumption, right, bad. None of this is really new. On the other hand, there are certain trace minerals and vitamins, things that have to come from the outside that we can't produce within our bodies. Requirements for those go up. So things like folate, fish oil is good to supplement, helps brain growth, right? But all of these things that I've shown up here are all found in fairly trace quantities, and they have bioactive effects. If we're talking about birth weight and fetal nutrition, that's a different story. That's actually about building a baby, right? That's about protein, fat, sugar, macronutrients. And so what do we know about macronutrient intake and total calories, protein, fat, carbohydrates during pregnancy, the mother's intake of macronutrients and effects on birth outcomes? Now, for decades in public health, there have been interventions and trials where they've supplemented the diets of pregnant women to see if they can improve birth outcomes, especially in populations that are undernourished. And generally speaking, they don't succeed. They have very small effects. So this is a meta-analysis where they combine all of those findings in one place. And in adequately nourished women, there were 5 studies. The effect of the supplement is on the order of an ounce. So that's the difference between giving birth to a 7-pound 7-ounce and a 7-pound 8-ounce baby. Very minor effect, and it wasn't even significant. You would expect a bigger effect in undernourished women, and you do see a slightly bigger effect, but it's less than an ounce more than that. Okay, and it's still not quite significant, but pretty close. And I should mention that these supplementation trials, they're not just nudging diet. They're actually providing between 500 and 1,000 calories during pregnancy. So they're pretty substantial. And they're really not having effects on birth outcomes. So, yes, the first 9 months in utero do have formative effects on our health long term. And nutrition is clearly an important part of that. We know that. But at least with respect to macronutrient intake, it doesn't appear to be the case that it's what the woman is eating during pregnancy itself that matters. And so this leads to a new question, and the one that I'll be exploring today. Might a baby's birth weight be predicted by the mother's history of nutrition during her own development? And this is one of the questions that we're exploring in our research in the Philippines. A study that's been going for 3 generations, over 30 years. I'm going to tell you about that, give you a little overview of that. And then we'll look at a few research questions. First, I've shown you some evidence that lower birth weight is associated with cardiovascular risk in places like the UK. Do we see something similar in the Philippines, a place where there's not as much overweight and obesity and where cardiovascular disease is a relatively recent phenomenon? We do, in fact. I'll show you that evidence. Given that, how might we go about trying to improve birth outcomes as a way to improve and lower cardiovascular disease risk? And we'll look at some evidence for that. And then finally, if a mother's early life developmental experiences influence birth outcomes in the next generation, that suggests that there's a memory that's lingering, right? That somehow that early nutritional experience persists and impacts the next generation. So I'll show you some of the work that we're doing that's aimed at trying to understand the mechanism of that memory. And then finally, we'll get to the evolutionary medicine question at the end. We'll think about all this stuff and what it implies about the potentially adaptive nature of some of these effects and what all this means for intervention design. Okay, so that'll be the wrap-up. All right, so the Philippines rains a lot. Big place, 100 million people. It's really a much larger country than I think most recognize. And it's about 1,000 kilometers across, so it's a big place. And over the last couple of generations, you've been seeing changes there, much like you see in other countries with similar economic circumstances. Infectious diseases are on the decline, and as a result, you're getting a rise of chronic degenerative diseases as the primary causes of death. In 2010, here you see the 10 leading causes of death. Heart attack is the first, but when we look at all the cardiovascular diseases, which also include stroke, hypertension, diabetes, kidney disease, it really dominates mortality there, much as it does here. Okay, so my research site, a place called Cebu City right in the middle of what's the central Visayas region of the Philippines. That horrible typhoon came right through here. They were luckily spared most of that. The study population, very diverse. So it's scattered across a big metropolitan area. And I should mention that Cebu City has about 3 million people, so it's not small. It's the second largest city in the country. And it's spread across a coastal area up into the mountains, rural to very urban. So it's a very diverse population. A lot of nutritional changes occurring there as elsewhere. Some key collaborators, UNC Chapel Hill has been there since the beginning when the study began in 1983. My own institution of Northwestern, and then the Office of Population Studies, which is located in Cebu itself. And here are some of the folks who have worked on this study. Some of these researchers have PhDs and they've been working on the study for literally 30 years. So it takes a big team to keep it going. The study's pretty remarkable. It started in 1983 with the enrollment of more than 3,000 women who are pregnant. And the offspring were enrolled at birth, and the original focus was here in infancy. It was all about infant feeding. What do you call it? Breast milk versus formula feeding. God, I just had a mental blank. And whether or not that impacts survival. So at the time, you know, certain formula companies were promoting formula, and they were having deleterious effects on growth. So this is one of the studies that originally showed that. A lot of detailed nutritional information back here. And then they followed them up to the present. I got involved in the study here in 1998, and I've been involved with it since. So it's an amazing record. So if we're interested in looking at some of these questions related to early life environments and their long-term effects, we can measure cardiovascular disease here, various risk factors, and relate that back to this nutritional history going all the way back to when they were actually in utero, prior to their own births. And so I'm going to start just by simply replicating what I showed you for the UK. Look at birth weight in relation to adult cardiovascular disease risk factors. And at this point, they're about 22 years of age when we're measuring this. So research question 1, at Cebu, do lower birth weight individuals have higher CVD risk as young adults? And here again, we see insulin. This is fasting insulin. As you would expect, individuals who as adults are heavier, who have a larger waist circumference, have higher insulin, suggesting higher risk of diabetes. When we cut this again and by tertiles of birth weight, you see that birth weight doesn't matter much back here. It starts to matter a little bit more here, and it matters most here. And the bottom line is that if you are lowest birth weight, these white bars, but you end up in the heaviest adult category, that's the highest risk scenario. And the difference here is about 25% across these birth weight tertiles. So it's actually a pretty substantial effect, especially among those who end up gaining weight as an adult. All right, so that's the basic story that we see, and it's similar to what other populations have been found as well. So we see evidence for lower birth weight being associated with higher blood pressure, cholesterol, inflammation, insulin resistance. Right? So it's a very similar story to what you see in places like the UK. Now given that fetal nutrition and lower birth weight seems to elevate risk for these diseases in the long run, what might be done to elevate birth weights and thereby improve health in cardiovascular health? If lower birth weights increase adult CVD, which maternal nutritional factors predict the weight of a baby at birth? And I've already mentioned that I'm going to be looking at nutritional history, the mother's history prior to pregnancy. We can't actually look at that in the original birth cohort, okay, because those women were enrolled when they were pregnant, so we don't have information going back prior to the births. What we do have is the births of this third generation here. The offspring are about 32, so many of them have been having kids for 10 years or more. And so now we can look at birth outcomes here in relation to this history. Now, let me just show you. Here's one of the original mothers who was enrolled when the study began. Here's her daughter who was in utero when the study began, and then granddaughter who was born more recently. So it's all 3 generations. Now, since 2009, we have had funding, in part through NSF, that's allowing us to track new birth outcomes. So we've been measuring diet and getting blood spots and all sorts of things during pregnancy, and then we measure birth weight, and we're going to be able to use that to do pretty, I think, good analysis of nutritional predictors of birth outcomes here. We're still collecting those data. We don't have the final data yet. We're going to end that probably in about 3 months. So what I'm going to present today are pilot data. This is recalled birth weight measured in 2005. We're going to be relating that to the mother's nutrition during adulthood. Because it's recalled, we don't have diet during pregnancy itself. But we can kind of say roughly, what is the nutritional status and nutritional intake of a mother as an adult, roughly. And then we can relate it back to her early life nutrition as measured back here, and even to the grandmother's nutrition when the mother was in utero. So that's the kind of framework we're going to look at here. And the question, what aspect of a mother's nutritional experiences, present or past, predict offspring birth weight? We'll start real simple. Okay, here we just see the association between the size of the mother at birth and the size of the baby at birth. Each kilogram increase in the mother's birth weight predicts about a 300-gram increase in the offspring's birth weight. Now, clearly, a lot of that is probably genetic. And if it was all genetic, we would expect that a 1 kilogram increase in the father's birth weight would similarly have about a 300-gram change in the offspring, because you inherit roughly 50% of your genes from each parent. There are some exceptions like mitochondrial DNA and Y chromosome, but this is what we find. The association is about half as strong. Okay, so something like mitochondrial inheritance could be part of this, but it's also potentially consistent with an intergenerational effect of fetal nutrition. So that's one idea, and that's actually been hypothesized, starting back in the 70s and 80s in some of the public health literature. So we've got some evidence for that here. Now let's look at actual dietary intake, caloric intake. We're going to start with the grandmother during pregnancy. Notice that there's a lot of scatter here. As a nutritionist, you would recommend that we would measure diet across 3 or 4 days and take the average of it. But back in 1983, when this study began, they just took a single dietary assessment. So it's pretty noisy data. But despite that, you can see that we're picking up a significant signal here. So the more caloric intake of the grandmother during the pregnancy of the mother, the larger the grand offspring's birth outcome. And this is from a fully adjusted model that includes the mother's adult size and her caloric intake and so on. I'll show you that at the end. This could be telling us something about sort of the socioeconomic or nutritional characteristics of that household generally, right? That might be sort of a general marker of that rather than something specific to pregnancy. To evaluate that, we can look at the mother's caloric intake in the postnatal period at 2 months of age. When we do that, you see that there's really no association. Okay, so it appears to be really specific to the grandmother's pregnancy when the mother was in utero. All right, now we can look at the mother's intake at various points in her life cycle. So far we've looked at the grandmother. So here's the mother's own intake when she is an infant, very similar, positive association, meaning that if she consumed more at about 2 years of age, more caloric intake, her baby ends up bigger 20-some years later, about 20 years later. We can also look at her intake in adulthood, and here we actually are averaging over multiple days, so this is more reliable data, but despite that, no association whatsoever. Okay, so the mother's caloric intake in adulthood doesn't matter, but her caloric intake in infancy does. And I've already shown you this evidence that the grandmother's intake during the mother's pregnancy matters. All right, so this is what's going on. Down here we have the current generation that's being born, and we find that the mother's intake in adulthood doesn't matter, but really what matters, it seems, is these measures of nutrition earlier in her life cycle. Right? And here's the final model. This is adjusting for sex of offspring, gestational duration, parity. Here are the 3 things that I just showed you. We also looked at other things that I'm not actually going to show you the data for. One is diarrheal morbidity during infancy. This is one of the primary causes of nutritional stress in this population, of growth faltering. More diarrheal morbidity, you see smaller baby in the next generation. Breastfeeding duration and exclusivity. These are both protective against diarrhea, and they also have positive nutritional effects during infancy. Those are associated with better birth outcomes in the next generation. These were not quite significant, but they were in the right direction. And then with adulthood, this is the thing that's kind of shocking, is that these really did not shake out. The mother's caloric intake in adulthood, her income, education, urban status, etc. Now, I don't want you to take away from this that I think that each of these is actually causally related to birth outcomes. This is a pilot analysis, and the main point of this is that when we look at adult characteristics, they don't seem to be important. When we look at earlier developmental and even intergenerational nutritional characteristics, they do seem to matter. So that's kind of the initial story that's emerging from our data. Let's look at this through a different lens. We're going to move up into the north of the Philippines to a place called Antipolo City, which is outside of Manila, about 15 miles. There it is. It's in a rural area, mountainous, and this happens to be the Shalom Birthing Clinic, provides services to lower-income families. And the sample here is 84 women who delivered singleton newborns in the summer of 2012. Now, this was the summer of 2012 because this happened to be a study conducted by a very ambitious undergraduate who worked with me. He had a connection to this clinic and he wanted to conduct a study. We designed it. He went off and he did an amazing job. Green Chung is his name. He's now going off to med school. But he worked with the midwives at the clinic and did a great job with this study. Now, we were interested in the same question. Given that there's a lot of low birth weight in this population, what are the predictors of that? Can we figure out some way to evaluate the impact of the mother's early nutritional experiences as an influence on birth outcomes? Now, in this sample, we don't have the 30 years of data that we have at Cebu, so we had to be a little more creative. And I'm going to explain the logic of what we did here. We hypothesized that the mother's leg length would be a stronger predictor of offspring birth weight than will her trunk length. And the rationale here is that leg growth is fast and it responds to nutrition during infancy and early childhood. So when you see secular trends through time where children are taller than their parents and parents are taller than the grandparents, that's generally speaking, that's about leg length getting longer and responding to nutrition. All right, and whereas trunk growth, on the other hand, is slow, it's less nutritionally sensitive. You can think of it as maybe more genetically canalized. So if we see a stronger relationship between leg growth and offspring birth weight than between trunk growth and offspring birth weight, that would be indirect evidence that there's an intergenerational effect of that woman's early nutrition on the next generation. And that's what we find. So here's leg length here. So what we're predicting here is we're just showing the percentage of variance explained. So if you look at birth outcomes, if you look at birth weight in this population, there are a few heavy birth outcomes and there are a few light ones and there's a lot in the middle. There's a basic bell curve. Of all that variation, 15% of it you can explain just by knowing the mother's leg length. That's about 7 times as much variation as explained by knowing her trunk length. Okay, so quite a bit stronger association. We also looked at placental weight, with the placenta being the interface between the mother and the fetus. And again, we see a strong relationship with leg length and essentially no relationship at all with trunk length, completely flat. All right, so what we think is going on here, the interpretation is that women who are as young girls grew better, okay, because of more favorable nutritional environments and or less nutritional stress, grew longer legs as a result of that. They go on to have babies that have higher placentas and larger birth weights as a result. And when you look across tertiles of leg growth or leg length in the women, see there's about a 300-gram gradient, so it's not trivial. Some of this is genetic, certainly, and some of it traces to other factors, but I think there's some hints here that there's an effect of nutrition as part of this. Okay, so far, Cebu and Antipolo, what we see is that measures of the mother's early nutrition, developmental nutrition, predict birth weight of the next generation. And so this leads to, I think, an important question, which is the third question: where's the biological memory of these early experiences? Where does it persist and how does it manifest? And one good candidate mechanism that's getting more attention in this literature is epigenetic changes. So epigenetic modifications, these are changes in chromosomes that leave the DNA intact. So if whatever genes you inherit from your parents is not modified by this. What is modified is which genes are expressed and in which tissues. And you can either amplify or silence genes through these mechanisms, so that you can kind of think of them as volume controls on genes. And importantly, some epigenetic settings may be modified by early experience and then persist into later life. So it's a good candidate mechanism for memory of early experience. One particular mechanism that's gotten most attention, at least in the human literature, is DNA methylation. And this basically involves the attachment of methyl groups to these CPG sites here in the double helix. Although this is not always the case, but if you have more methylation in the vicinity of a gene promoter, that tends to silence gene transcription at that gene, right? So you can look at levels of methylation and evaluate this. So excellent candidate tissue in our case, again, is the placenta, because it's the interface between the mother and the fetus. You can imagine that genes that regulate things like nutrient transporters or hormones that regulate growth, all of this could be modified potentially by her early experience, and this could manifest in altered placental function. And that's basically just stating what I'm saying here. So, all right, so the pilot study, we got some NIH funding to conduct this. This is very hard to do. And if you have a community-based study where everybody goes to the same clinic and gives birth, you could just kind of set up there and wait for people to come through. But in our case, this is a population representative sample that's spread out across 3 million people. And so everybody's going to different clinics and in hospitals. So what we did is we isolated some of the more heavily used hospitals and set up in each of those and got people trained. And we managed to get 22 placentas during the period of this small study. We collected tissue for both anatomical and epigenetic analysis, and this is with my collaborators Julian Rutherford at UIC, Derek Wildman at Wayne State, and Nanette Lee and others from the Philippines. All right, and so the approach here is pretty simple. We have a very small sample size. All we wanted to do is look at whether or not patterns of methylation were different among individuals from the lower versus upper half of the mother's height as a very crude measure of that woman's nutrition. Okay, we don't have leg length from Cebu yet. We're going to get it next time. So we're just using her height generally as a crude measure of early nutrition. Whether methylation patterns vary on the basis of placental weight and also birth weight. And so let's look at that. How many genes are differentially methylated across these 3 different phenotypes? And here's what we find. 591 mother's height and so forth, quite a few. Now what's interesting is when we look at the intersect here. Okay, these genes right here that are differentially methylated are differentially methylated both in relation to the mother's height, which is a measure of, kind of a crude proxy of her early nutritional experiences, but also in relation to birth weight of the next generation. So those are the kinds of genes, those 77. There might be some in there that are, that actually link the two, right? That are impacted by the mother's early nutrition and then go on to influence birth weight in the next generation. So that's the idea. That's the kind of approach that we're doing here. It's a simple analysis, but we hope to scale it up. And so thus far, we're seeing some interesting initial findings with pathway analyses. See overrepresentation of morphology of the placenta, folding of embryonic tissues, morphology of pancreatic cells, mTOR signaling pathway. So this is something that's very much instrumental in embryonic growth. It's important in early growth. Insulin signaling pathway. So to the extent that those differential patterns of methylation relate to early nutrition, which we don't know yet, but we can clarify in future research hopefully, these might be interesting mechanisms to explore to underlie these effects. Okay, so let's go ahead and recap some of this. So we started with the evidence that lower birth weight is associated with higher cardiovascular disease risk in the Philippines, much as you see elsewhere. Now, given that then, what are the predictors of birth weight? What are the potential levers that could be used as a public health tool to improve health over generations? And generally what we're finding, and all of this is pretty preliminary, but that measures of the mother's early life nutrition seem to be more important as predictors of offspring birth weight than are her adult nutrition. At Cebu, we see this in evidence that the grandmother's nutrition during the mother's pregnancy, the mother's fetal and infancy nutrition predicts offspring birth weight, whereas the mother's nutrition as an adult does not. Okay, so that's interesting. And then at Antipolo, we don't have great measures of the mother's adult nutrition. We had the body mass index, which was not a strong predictor, but we find that leg length is a stronger predictor than trunk length, consistent with some effect lingering of her early infancy and childhood nutritional experiences. All right, so what does this mean for public health, briefly? What this shows here are the mothers of the original cohort who were enrolled when they were pregnant. And back in 1984, after pregnancy, only about 5% were overweight or obese. But as you can see over the next 14, 15 years, that increased to about 40%. And their offspring who were lean back in 2002 when we measured many of those cardiovascular risk factors are gaining weight, and they're going to continue to gain weight as well. And what I think our findings suggest, and findings in other studies as well, is that as those individuals gain weight, it's the ones who were born lighter, okay, suggestive of fetal undernutrition, who are going to have more adverse increases in cardiovascular disease as a result, right? Now, as a public health question then, what can we do about that? One strategy I think that our data are starting to point to, as are other studies, is that improving the nutrition and growth of future mothers, that is the young infants and children of today, is an important means by which you might reverse some of those effects. It's not enough to simply supplement during pregnancy. You got to worry about actually altering development in the next generation as a long-term strategy to improve health in future generations. All right, so I've given you evidence now that the fetus is responding to nutrition, it's modifying its biology, and it's doing so across a range of different systems, and some of those are quite important. biologically and in terms of adaptation. And ideally, we would go beyond just kind of describing these associations and have a better understanding for where they came from and whether or not they have a function. And that's what I want to kind of dig into in the last bit of my talk here. Why do our bodies modify our biology in response to prenatal markers of nutrition? Is just this sort of an accident or effect of kind of damage in utero that's lingering long term, or is there something more? And if there is something more, what does that tell us about our best approaches to public health? And one idea that's gotten a lot of press, and I'm going to go back to this article as an illustration of this, is that there's more than just resources being conveyed between generations. There's actually communication, there's information. Here's Annie Murphy-Paul again. The fetus treats these maternal contributions as information, biological postcards from the world outside. What a fetus is absorbing in utero is the answers to question critical to its survival. Will it be born into a world of abundance or scarcity? And here she's channeling people like Pat Bateson, who've talked about the idea that the mother is sending a weather forecast to the fetus, allowing it to adjust its biology in anticipation of scarcity. Gluckman and Hansen very similarly talk about the predictive adaptive response idea. Now, many of you might be wondering, what's the benefit of setting yourself up for diabetes or hypertension or stroke, right? There's no obvious adaptive benefit to some of these outcomes that are associated with these fetal adjustments. But if you look at the underlying biology that eventually leads to those diseases, they could in fact be adaptive. So I talked about insulin resistance as something that's induced by prenatal undernutrition. What insulin resistance involves is muscle taking up less glucose. What that does is effectively allows more glucose to be shunted elsewhere. So during normal pregnancy, insulin resistance goes up as a way for the mother's body to shunt glucose away from her own tissues towards the fetus. Okay, that's David Haig's paper from 1993 is a wonderful kind of argument about that. Similarly, the brain can be a beneficiary of that, as can the immune system. So that you can imagine that tweaking the body's priority with where it's using glucose could have adaptive value, depending upon the type of environment you're going to be born into. So if you'll just humor me and take at face value for a second the idea that some of these biological adjustments, even if they lead to disease in a modern environment of chronic abundance and high stress and so forth, that maybe they have adaptive origins. Then the next question becomes, what information is actually being conveyed? Is the mother providing information to the fetus that's reliable and will allow it to make these kinds of adjustments in a way that makes sense? And here, we can just reframe this. What does fetal nutrition, in the case of nutrition, what does fetal nutrition allow the fetus to see about the environment that it will be born into? And here I think our data actually speak to this, as do other data. If the mother is consuming a bunch of calories and you don't see a change in the fetus, I'm going to assume that there's not much communication going on there. Whereas if the mother consumes a lot and there's a change in the fetus, there might be some communication. So if we just take that very simple approach, we've already talked about the fact that the mother's diet during pregnancy itself, it does have an effect on birth outcomes, but it's pretty attenuated, it's pretty small. There's a literature that I haven't talked about that goes back in public health, talking about things like pregnancy weight gain as being a strong predictor. That's kind of a no-brainer in part because you're really measuring the weight of the fetus in part. That's what that weight gain is. Paraconceptional nutrition and pre-pregnancy weight have also been known to be important predictors of birth outcomes. So this kind of reflects the mother's nutrition going into pregnancy. Then we have our findings. Infancy and childhood nutrition seem to be important, as does potentially the mother's own fetal nutrition and her gestational environment. Once you take this back to the mother's own fetal nutrition, of course what we're really doing is taking it back to the grandmother's nutrition. You can kind of repeat this whole process in the grandmother's generation. And so to the extent that fetal nutrition conveys information to the fetus and provides information about the ecology that it's going to be born into, it seems to be doing so on a variety of time scales. Right? Everything from days as a fairly weak signal, perhaps all the way down to decades or even generations. And so you can think of that stream of nutrients as a record of the matrilineal history of nutrition, more so than what the mother is experiencing during pregnancy itself. All right, so if that's the case, you may be wondering now, if the fetus is trying to adjust its biology in a way that predicts the future, in a sense, sort of anticipates where it's going to be, isn't it just receiving outdated information, all this historical information about what the mother experienced and what the grandmother experienced? Well, I'm going to argue that the benefit of setting fetal nutrition to average nutritional experiences over these longer time scales historically may relate to the unpredictable nature of natural environments. And here you see rye production. So this is just a very crude illustration of how unpredictable production can be within an ecology. So these spikes here reflect volcanic events. When there's basically very little sunlight and production goes way down, the price goes up. But the bottom line is that you can't really predict where things are going. There's a lot of variability, a lot of spikes, a lot of noise, even though there are some underlying trends. And I want to show you some data that are kind of arbitrary that illustrate a point and I think illustrate the function of this kind of effect that fetal nutrition has. So these right here, these are actually from an ice core. These are totally irrelevant data. But if we connect the dots, the main point is that there's a lot of variability, and if you squint your eyes a little bit, you can see that there's an underlying trend that goes through them. And you can picture this as being something like the productivity of the environment. How hard is it to get a calorie out of the environment, something like that. Now, instead of connecting the dots, let's start averaging. And we can start by averaging over 10 time units, and we can do it by 20, 40, 100. And the data are completely arbitrary. The units are arbitrary. The point is very simple. As you widen that window of averaging, you cut through all the scatter, you cut through the noise, and you can identify a more stable underlying trend, this dark blue line here. And this is what I think fetal nutrition may be approximating. It's buffered against transient short-term uptakes, ups and downs in the mother's intake during gestation. You can think of this as all that noise, season to season, good year to bad year, and so forth, right? All the short-term variability. Longer duration averaging allows tracking of stable nutritional trends over longer time scales, something like that more stable underlying signal, the blue line in the last graph. The result is that fetal nutrition may in fact calibrate to average recent nutrition, a more adaptively relevant cue for adjusting long-term metabolic and biological strategy. And I've called this intergenerational phenotypic inertia because it's intergenerational, it's information that's not genetic, it's clearly phenotypic information. And the concept of inertia reflects the idea that the phenotype in the present is kind of anchored to these past experiences, that it's not responding rapidly to what's happening in the present because it's being influenced by this inertial effect of the past. Now we can think about this in terms of the classic framework of human adaptability, where you've got ecological and or environmental changes that are occurring on the order of seconds to seconds down to very long time scales. And at the slowest level of adaptation, you have natural selection pruning through the gene pool and improving the gene pool. At the other end, we have physiologic processes and metabolic processes that are governed by homeostasis. And the key to homeostatic processes is that they are maintaining stability internally. They're like shock absorbers, right? They reverse the effects of the environment and maintain something that's fairly stable inside. And that can help probably all the way down to months and years. But you've got this gap in here of decades to longer. And these kinds of homeostatic processes can be quite effective, but they have limits. And an illustration of that would be moving to high altitude. So if you move to Nepal or something like that, your physiologic, your immediate responses are going to be homeostatic. You're going to increase your heart rate. You're going to start breathing more rapidly to try to compensate for the fact that there's low oxygen pressure. But that's not a good strategy for the long haul, right? I mean, that's a fairly inefficient way to adapt over more than a short period. If your kids are raised there, they get around that by growing larger lungs. Okay, and we know this they grow much bigger lungs. And so that's an example of developmental plasticity. The body responds to the environment by actually modifying its structure in a way that's adaptive to those local conditions. Right, now, the examples I've shown you, okay, of prenatal nutrition and the effects of mother's nutrition on the fetus clearly are examples of developmental plasticity. They're not genetic effects. They're the physiology responding to the environment in ways that are relatively durable. But they're fundamentally different than this example that I just gave about high altitude adaptation, because unlike the example of the lung, which is like literally interacting with the environment itself and responding and modifying structure in real time as it senses that there's low oxygen, what's happening in these other examples is responding to cues of past environments. You're not responding to the environment itself, you're responding to cues that reflect experiences of ancestors going back a generation or two. And it's that integrative nature, that more stable nature of that signal that I think allows a stretching of the utility of plasticity down into these longer time scales, okay, through an intergenerational process. All right, so this gives us some ideas then about supplementations and why they're rarely effective. If this is right, fetal nutrition may in fact be designed to minimize the response to transient changes in the ecology. One-time pregnancy supplementations may resemble just such a short-term unusual spike in nutrition, noise. And going back to our chart, maybe this is what a supplement looks like. Clearly would not make a lot of sense as a strategy to set your biology here, given that the actual trend is down here, and it may even be trending downward, right? So it would make a lot of sense to ignore that. Another way to think about this is in terms of the timescale idea. If you're modifying a pregnant woman's diet over several months, for instance, that's a typical supplementation, you're clearly in the green part of this chart, which means that you're pushing against homeostatic mechanisms mostly. Things that are changing on the order of months, our biology should be designed to ignore rather than incorporate and alter strategy for life. It makes very little sense given that we're going to live 70 years or however many years. The biological systems that we want to change are down here. Okay, that's, they should be cueing into things that suggest that there are durable changes in the environment that might last an entire generation or even more. And so that leads to my final point, a question which we can also discuss. How might we devise interventions that trick bodies into perceiving sustained ecological improvement? And I think that's a question that comes out of this framework. And that's all. Thanks. Looks like the Olympic torch.
Are there any questions? John? Oh, wait. So I was just wondering, in your analysis of impact, things that predict infant birth rates, you have the mother's adult diet, but you don't. Do you know about her diet during pregnancy?
Yeah, that's why I pointed that out. We don't know. No, we don't know because those are recalled birth outcomes. So what we know is
grandmother's diet during pregnancy.
Right, because that's when the study began. All the women were pregnant, and
Got it.
we are currently collecting.
Got it.
We are collecting the data that you're asking about, but
Okay.
we're not done, and that's why I did the pilot analysis.
Yeah. Okay.
So, but nonetheless,
Yeah,
I mean, it's
I take
diet.
your point about
we don't
the
expect
argument.
it to be bouncing around too much.
Yeah. No, I
So,
take your point, but I was just
yeah.
wondering if that helped or
Yeah,
or
I mean,
didn't,
it'll be
or
interesting to see if that holds up once we actually get the the dietary data during the pregnancies
You have
itself.
a strong prediction here, right?
Yeah, sure.
So that's cool.
Oh, sorry.
So I have a question, a comment. The question is, so it went by a little fast, and so you have strong evidence of a lagged indicator, so that the diet during the early life experience of a baby affects that when that baby becomes a mom.
Mm-hmm.
That's not an average yet. To be an average, it has to be the sum of a bunch of measures. And at the very end, I thought maybe you showed us evidence for that. But so do you have evidence that it really is not just a lag? Because otherwise, it's just a noisy indicator from 50 years ago, not an average, right?
Yeah, right. No, that's a good point. It's not, maybe average is the wrong way to state what we've found, but you can tell me. What we're showing is that there are independent positive relationships between her diet or her dietary history going back, a generation plus. So the
At
fact
different
that
times during
they're all
that.
independently related,
And
I guess I intuitively interpret that as something like an average, but
well, if
maybe
they
it's
kind of add together, it
Yeah.
is like an average. It
they're
depends.
additive and they're independent.
Yeah,
So
okay.
I interpret that as an average, but you're right. It's not. I'm not technically calculating.
The smoothing will
average.
come from the summing, not from the lagging.
Right.
Yeah,
Absolutely. The
yeah,
lag
yeah.
will just create
Okay,
maladaptive, interference.
The comment / question is so the question is, what is the fetus and the developing child using this information for, right? So the first thing I would think of is there's some life history trade-offs that we want to build a kind of cheap, if it's a lousy environment, we build a cheap baby that's not going to last very long and get things going, and if it's a great environment, we want to build a baby
Yeah.
a baby that'll last, right?
Sure.
But if that's the case, then you really only need to predict the next 5 or 10 years or 15 years, right? Because after that, it's, you've already done all the building and it's too late. So that makes me wonder so you really need to know, in whatever the ancestral environments are, what lags are going to predict the first 10 years or first 15 years, if that hypothesis is right, the first 5 or 10 or 15 years of the child's development, right? And it seems like going back to grandparents is a pretty long lag to me, and maybe you'd really be better off paying attention, if you're natural selection, to the mother's current nutrition, right? Because that. So, I wonder if you've thought of any. I mean, it also occurred to me that another hypothesis you might have is that in some social systems, babies get treated really badly. In other social systems, they get treated better, and that the mom's experience as a baby would be a better predictor of the social system you found yourself in than the mom's current nutrition, maybe. I don't.
Right.
So,
Okay.
so in patrilineal societies, often moms get pretty short shrift, whereas in matrilocal societies, moms get treated better, et cetera.
Yeah,
So
those
I'll
great,
shut up now.
great. No, those great questions. I'm not sure that the adaptive relevance of body size ends when growth ends, right? I mean, because what we're really talking about here is a kind of trajectory of development that continues to have implications for caloric requirements and energy expenditure across the life cycle. So I agree that maybe where the rubber meets the road, in terms of strongest selection, is probably going to be during infancy and childhood. But I think that there continue to be differential costs and benefits and so forth, of being larger or smaller as an adult, right? So I don't know that, I hear your point, but I think that there probably could be longer term implications. The second point was about, remind me quickly, sorry. It was basically about social stress as opposed to nutritional stress.
So
Yeah.
your hypothesis
Yeah.
is a smoothing hypothesis. If childhood environments
Yeah.
are correlated from one generation to the next, then you might care a lot about.
Yeah,
the
absolutely.
mom's childhood environment.
And really, I think there's a whole range of interesting issues that come out of this question, because I've shown this chart here that shows this supposed natural environmental variability as if, we're out there alone, just kind of experiencing the environment by ourselves. What we're really doing is we're part of these complex social units that have all sorts of reciprocity and buffering mechanisms themselves, right? And so the nutritional experiences that drive early growth have a social basis to them. And if that social fabric is broken down, then that's going to impact the nutrition that you receive, presumably, right? Because the whole system is based on cooperation and reciprocity. So I think that there could, part of my answer to what your question is that part of what the nutritional signal is about is probably the cohesiveness of that social unit, which could change through time. The second point about psychosocial stress and its impact, I don't think that the scenario that I laid out here could really work for that because of the biology of stress, which directly impacts the fetus immediately. So if you're experiencing stress, just during a fairly narrow window of late gestation, your stress physiology is actually altered for life as a result of that. So a chance event can have fairly long repercussions. I don't see that as evidence of an adaptive adjustment. It doesn't seem plausible to me because it is so transient. So, yeah.
I'm sorry, Kim.
Okay, I might be returning a little bit to one of Rob's points, but maybe I'm going to say it differently. I guess, and I don't know the actual biological details, but my first kind of question about everything is, do we know who's controlling birth weight? Could be that mom is controlling birth weight, and it could be that the child is controlling its birth weight. And it seems like we got kind of an adaptive story of the way the child should be designed to phenotypically, adaptively react if the child had control over its birth weight. But what if it's mom that's controlling the birth weight? And so the real issue is mom experienced stress as an infant and had poor nutrition, and therefore mom is now adjusting what the birth weight of her child is going to be as she produces children based on her experience, and it gets, you know, into other possible explanations about how much you would invest per child as a function of the stress you went through as an indicator of the environment again, and so on. So I guess without talking too much, who do we know who's
Yeah.
in control of birth weight? And it's a really different adaptive story if mom is the one that's adapting to things in order to produce a birth weight than if it's the child that's using signals from previous generations
Right.
to determine its birth weight.
Yeah, it's a great question. The simple answer is we don't know. The complex answer is that it seems to be both. So certainly maternal glucose levels, for instance, her circulating glucose levels are directly related to the birth size of the baby. But the mother's circulating glucose levels are in part a reflection of fetal and placental manipulations of maternal biology. So there's some fairly complex scenarios that are going on with genomic conflict. I mean, you've seen Haig's work, I'm sure, on this. There's another twist to that. A lot of those imprinted loci that drive the genomic conflict are themselves seem to be responsive to early nutrition. So the degree of silencing, parent of origin effects is not all or nothing. It's actually modifiable in response to early nutrition. So there's a lot going on there, and I don't have a good answer to your question yet. I mean, I think the mother certainly sets an upper limit on birth size because of the size of the birth canal. But with respect to the metabolic drivers of growth, it's the mother, but it's also the fetus in response to the mother. There's a lot of complexity going on there. I think that your point is going to be most relevant for if you had a purely monogamous species, it wouldn't matter too much. But for a non-monogamous species, it comes to matter. I mean, that conflict between the maternal and the paternal interests within that baby become an issue, and yeah, so we're somewhere in that spectrum, and I think we're we're closer to the monogamous spectrum than a lot of the work on imprinting and genomic conflict that it came out of mice and species that have a lot of conflict over resources. So I don't know if I answer your question. I don't have an answer to it. That's probably why. But I think it's all of the above. I think it's the mother and the fetus, and we don't really know yet who is in the driver's seat. Nor do we know who is exactly in the driver's seat of actually ending pregnancy, because that's an interplay between the placenta and the mother and the fetus. There's a lot going on there as well.
So Chris, my question is just a variation. I'm curious about why these physiological systems get locked in for a lifetime. And it seems to me that, the big decision that has to be made is how big is this baby going to grow up to be? That has to be over the long term.
Yeah.
And I think you can make an argument, or you have, for the significance of longer term data to make that decision. But why lock in all of these other systems in a way that they're stuck for a whole lifetime?
Yeah.
I mean, the very variation that you showed would seem to me to argue that it would be very dumb for natural selection to lock in insulin and all the rest for a lifetime. It should change from year to year.
Yeah, well, I think it does. I think that I think all systems change on multiple time scales just as they adapt on multiple time scales. I mean, it's sort of the baseline. If you are going to modify your insulin physiology in adulthood, where's your starting point? It's a starting point that was established in utero. So it's not that it's not modifiable, it's just that where you begin as an adult and where you start to modify from as your baseline is what I think is being modified. But part of the answer to your question is that there may not be an opportunity, there may not be a choice really. We develop once. Development starts with pure pluripotency, one cell that can become any cell type, and gradually it becomes a more differentiated, committed
It's
phenotype.
path dependence locks it in. Yeah.
What's that?
It's just path dependence.
Yeah, path dependence, right. So, I mean, gradually, there are certain decision points that are fundamental, and that you can't reverse. And so some of this might be about that. But I agree. And I think that we do see evidence that some of these systems change. But nonetheless, you're going to pick up the signal because the point at which you started to change is different, right? I mean, that's how I would think about it.
Hi, thank you for your talk. One question I have is related to 2 points that you brought up. One, the sort of weather predicting of the mother to inform the fetus of the environment that's going to be born into. And the other one is the disease, chance of disease risk. In previous courses I've taken, we learned a lot about the Dutch hunger winter and things like that in Britain and Europe, and they basically came to the conclusion that if the fetus was born into the same environment and grew up in the same environment that was predicted, they had a lower risk of diseases or less severity of diseases than if the fetus was born into an environment and lived in an environment that wasn't predicted by the mother. So I was just wondering if you had any indication of that from your research, or that's something that you're going to look into. I don't know about the difference in caloric intakes and things from your study population and if they're different or not.
That's a great question. I would like to see that evidence, because I've not actually seen, I'm very familiar with the Dutch famine study, and I'm not all that familiar with evidence that suggests that, you know, deprivation followed by deprivation is good for health in that context.
It's
But
not that it's good for health overall.
better.
That when the environment that was predicted didn't match what the
Yeah.
child grew up in, that the effects were worse.
Yeah. See, this is not going to answer your question, but it gives me an opportunity to make another point. One of the things that I think is really fascinating about the Dutch hunger winter. Okay, so this is one of the few kind of quasi-experimental tests of this idea that fetal nutrition has long-term effects in humans. And it was World War II. There was a Nazi blockade of a certain area of Holland, and so they had pretty good records on who was eating what, and intake went way down as a result of that. And now they followed up the next generation, and they've looked at a whole range of outcomes. They do see some epigenetic changes that persist. They do see some long-term effects, but I think what's really notable about all of it is how small the effects are. Okay, the effects of that very severe nutritional stress experienced by the mother during pregnancy did not have the big effects that we see, for instance, in animal models that are experiencing the same kind of magnitude of nutritional stress. That, to me, argues for the power of buffering in us compared to smaller, shorter-lived species. So, I mean, that's sort of what I take away from the Dutch hunger. As to your question, we don't have, you know, as of now, we haven't really thought about how to test that idea. The one example that I can think of where they've kind of evoked that idea is the Gambia, where there's big seasonal fluctuations in intake and so forth, and they're not finding associations between measures of prenatal nutrition and long-term disease risk, and they're arguing that it's because it's still a fairly deprived environment, so maybe that's why. Yeah.
Well, I think we're done. Thank you very
Okay,
much.
thanks.